Hello everyone, welcome to this presentation. We are now on part 4 of this series of lecture on protein. Proteins, remember they are a large molecule. They are composed of they are made up of amino acids. Since they have many there are many kinds of proteins, and their structure depend on the amino acid content. Their shape also is dictated by the amino acid content. Okay, and proteins are classified based on shape. We have we have fibrous, globular, and membrane protein. Okay, fibrous proteins, they are proteins with elongated shape. Usually they are insoluble in water. Okay, so one example of protein that belongs to this class is the collagen. Okay, so collagen is made up of triple helix, three three polypeptide chains that are intertwined with one another. Okay, next is globular proteins. These proteins are folded, okay, spherical or globular, and normally they are water soluble. The reason why since they are water soluble, you would expect that polar amino acids would be on the surface of the protein. However, the hydrophobic amino acid residues are in the protein core. Okay, usually the hydrophobic, since they don't interact with water, they usually are found at the core of the protein. Those at the surface, those are the amino acids that could interact with water. Therefore, these amino acids are the polar. Those at the core, we call them okay, non-polar. Okay, the third classification of protein is the membrane protein. Okay, and these are the proteins that usually are associated with cell membrane, and they must be insoluble in water. Okay, the hydrophobic amino acid residues are at the surface. All right, because they don't interact with water. They don't like water, and that's the reason why they are insoluble. What they do is they help in the transport of molecule across the membrane. Okay, so these are the proteins here. They serve as channel. Okay, we call them membrane proteins. As I said before, proteins have various structure. That's why they have various um, uses also or function. Okay, proteins do not only make your muscle grow, but they have more functions other than that. It says here the functions include the following: catalytic protein. Uh, some of the proteins or many of the proteins, they perform as catalysts. Okay, they have catalytic function, they have catalytic action, and those proteins that have catalytic, okay, action, they are called enzymes. Proteins do the defense, okay, in our body in the form of immunoglobulins, or what we know as antibodies. Some proteins, they could transport molecules in order to be transported to the different parts of the body. They are called transport protein. Okay? Say for example, the protein that transfers or transports iron, okay? The protein that transports iron is called transferrin. Okay? The protein that transports the protein that transports oxygen is called um, hemoglobin. Okay, Messenger protein, those proteins that transmit signal to coordinate biochemical processes between different cells, tissues, or organs. Okay, examples of protein that belong to this, you have insulin and glucagon. Insulin regulates carbohydrates level. Okay, when there's a increased carbohydrate in the, or glucose in the blood, okay, insulin mediates the or facilitates the uptake of glucose into the cell. So for example, this is the cell, okay? These are the receptors, okay? Receptors. They could be glycoprotein, okay? So when insulin binds to this receptor, um, a protein channel opens in order for the glucose to pass through. So insulin mediates, okay? Um, it's a messenger. It gives message that a glucose needs to be, or glucose molecules uh, need to cross the cell membrane. Okay? Um, Glucagon does the opposite. Glucagon does its work when the carbohydrate level in the blood is low. Okay? When the glucose level is low, okay, the glucagon will um, stimulate the release of glucose from the glycogen. Remember, glycogen is the storage form of carbohydrates. So when the glucose level drops, the glucagon will stimulate the breaking breaking of glycogen to release glucose. We have contractile proteins, okay? These are proteins that are needed for movement. We have two common examples. You have actin and myosin. We have also structural proteins that are responsible for stiffness and rigidity of the protein. Two examples, keratin and collagen. Okay, present in nails and in hair. We have transmembrane protein, transmembrane. These proteins span in a cell membrane and help control the movement of molecules and ions. Okay, so as we have seen in the previous lecture, this is the cell membrane. Okay, transmembrane protein, okay, penetrates the cell membrane, spans the cell membrane. Okay, these proteins serve as channel to regulate the movement or the movement of molecules across the cell, in and out of the cell. Storage proteins, they bind and store small molecules. Ferritin stores iron. Okay, myoglobin stores oxygen. Then we have regulatory protein. Okay, these proteins serve as receptor of molecules. Usually, they are at the surface of the cell. They regulate um, the communication between cell. Then we have nutrient protein. Okay, particularly important in the early stages of life. We have casein present in milk, ovalbumin present in the egg white. Okay, they are considered as nutrient protein. In order for our body to get benefit from protein, the protein must be hydrolyzed. Okay, because proteins are big, then it cannot be assimilated by the cell. So what happens is in the stomach, okay, and then into the small intestine, the protein would undergo breaking. What happens is it's called protein hydrolysis, or simply breaking of peptide bond. I have shown you earlier how peptide bond is formed. So protein hydrolysis sa process wherein peptide bonds are broken. So this is the these are the peptide these are the peptide bonds, okay, and another peptide bonds. Okay, so this is your tri this is the tripeptide, 3 amino acid residues connected by 2 peptide bonds. When these peptide bonds are broken in the presence of water, that's why hydrolysis, okay, the 3 amino acids are formed. Okay, alanine, glycine, and serine. Remember, the name of this tripeptide is alanyl, glycyl, serine. But when they are released as individual amino acid, their names retain. Okay, alanine, glycine, and serine. Okay, those enzyme, okay, hydrolysis or breaking of peptide bond in our intestine is catalyzed by enzymes. Okay, as I said earlier in the previous statements in the previous slide, enzymes are proteins that have catalytic action. So generally, the enzymes that catalyze hydrolysis of peptide bond, we call them protease. Okay? We call them protease. Under certain condition, proteins could undergo denaturation. Okay? In denaturation, it says here, denaturation is a process wherein the protein is partially or completely disorganized. Okay? The protein's tertiary structure is disrupted. Okay, here you have a normal protein that assumes its native or physiological shape. When this protein is subjected to denaturation, okay, the protein loses its tertiary structure. Remember, tertiary structure is governed by four forces. Okay, so it's only the in denaturation, it's only the uh, four attractive forces that maintain the tertiary structure is lost. Okay, but the amino acids remain intact, remain connected. Unlike in protein hydrolysis, of course, the protein will be denatured first, and then the amino acids are cut. The peptide bonds are cut in protein hydrolysis. But in protein denaturation, it's only the shape that is altered, okay? But the amino acids remain connected. Protein denaturation could mean loss of biological activity. That's why when enzymes are denatured, remember enzymes are protein, so when enzymes are denatured, that enzyme could not perform its catalytic activity anymore because of the loss of its shape. Okay. Now let's go to other kinds of protein. These are protein derivatives. Okay, and there are some of them. Glycoproteins. These are conjugated proteins. Remember, I said earlier, conjugated protein, they have prosthetic groups. Okay? Prosthetic groups are non-amino acid component. So conjugated proteins, the uh, non-amino acid is carbohydrates. Okay? Many of the plasma membrane proteins are glycoproteins. You can find them, you can find these glycoproteins on the surface of the plasma membrane. Okay? Lipoprotein, okay, this is another example of conjugated protein. Um, their function is to help suspend lipids and transport them through the bloodstream. And lipoproteins are classified based on component. You have chylomicron, very low density lipoprotein or VLDL. We have LDL and we have also HDL. They differ in the percentage of cholesterol, phospholipid, and triglyceride or TAG. These are the functions of each of the lipoproteins. Chylomicron transports dietary TAG, okay? VLDL transports uh, triglyceride in the liver to adipose tissue. Low density or LDL transports cholesterol in the liver to the cell. That's why LDL has been associated with bad cholesterol. Okay. HDL or high density lipoprotein collects excess cholesterol from the body tissue and bring it back to the liver for degradation to bile acid. So this is also known as the good cholesterol. Okay, but take note, we don't use that term as in science, no, good cholesterol and bad cholesterol, but we have only LDL and HDL. Okay, so as you can see in the table, they differ in the components. Okay,